Okay, so I will repeat a little bit what you heard already, but I'd like to add a little bit of emphasis on this workshop. So, we run this workshop since a long time, since 2003. And I personally taught, like in groups like you, over 1,000 participants. I think now maybe already 1,200. So I'm really very eager to do this because you develop tools, but you must also tell people how to use the tools. And so the training is really a very important part of making the software available. So if we distribute VMD and MD, we also want to tell people what you can do with it. And so one of my coworkers always tries to take nice pictures of the place where the workshops take place. And but you probably don't have too much time to run through Pittsburgh. Maybe in the evening, go to some nice restaurant. And we always show the group picture of the last workshop. So here we will take a group picture also today. I forgot when, maybe today or tomorrow. And this one here is actually the last workshop we did in Munich, in Germany. So that was just about 2 months ago, and was also a very nice, fun workshop. Okay, so you saw already who is doing the teaching, so I can be brief here. And I just would like to tell you about the program. That the program is divided into morning sessions and afternoon sessions. In the morning, we have normal classroom-style presentations, and we talk about a little bit, because there's not that much time, on the algorithms that are used in these simulations and computational strategies, and how to actually do practically the calculations. And in the afternoon, you sit down with your laptop, and next to it you will have a tutorial printed out, and then you work through the tutorial. And the tutorials we develop for self-study, so you sit there and you could do it also at home. You can do it this evening. If you don't get finished, you can take the material home and do it there. But in the setting of a workshop, it's nice because you can ask questions, and you also are a little bit more disciplined. You go more towards the end of the tutorial, where sort of like the more useful and powerful stuff is done, whereas if you just do it on a Sunday morning, you never get to the end. And so, the afternoon session is by far the most important part of this workshop. So the morning is just sort of to introduce you a little bit, get you into the mood, but the afternoon is important. And we have a good number of teaching assistants that cover the afternoons. And so there, you just raise your hand or look a little helpless, and there will somebody coming and asking what question you have. And it's also, however, nice if you help each other a little bit. That is sometimes a faster way of communicating, and so that could also contribute to the success. But please realize is that if you sit here doing the lecture in the morning and then don't really do much in the afternoon, then it's a total waste of workshop. That's why we call the workshop the hands-on workshop, because of the afternoon sessions. We can only do a few of the tutorials during this week. There are many tutorials on our website, also on the website of my colleague Yvette Baha here in Pittsburgh. And so you can download more tutorials on special areas. So once you get the taste of them, then you will easily see then, oh my God, this is something I could really use. And then in the same style of the tutorial that you became familiar with here, you will be able to train further on. Okay, so the Tuesday and next days all are the same. On the first day I introduce VMD and a little bit molecular dynamics, and the second day I talk about molecular dynamics more in general terms, not so much about particular systems, although at the end I give some overview of the recent work that is done in the field of molecular dynamics. And then my colleague Alek Aksimentiev will talk on Wednesday, and he's an expert in nucleic acid simulations, both on all atom and coarse-grained levels, and he's also interested in the entire genome, the mechanical properties of it, so it should be an interesting introduction to these systems, both on the small and the large scale. And then the Pittsburgh team takes over, and Yvette Barr is an expert in elastic network models that describe the key essential motions also over very long times of proteins, and they are really a great tool opening one's eyes about what these biomolecules are capable of. And so she introduces that subject, and then and then we also have then some lectures on, I think we got it here wrong, we must have, yeah, we must not, this should be Friday. So on Friday we have then applied areas that could also be interested for basic researchers, because it's always nice to look around the corner, because most of the time when you start your papers on some basic problem in molecular biology, you write in your introduction, and this work will be very important for finding new drug treatments. And so it's going good to know what these people are actually doing, and maybe actually one day you can relate your work to drug treatment. So I think it could be a very, very useful for you, even if you're not directly interested in this area. So here are some remarks I like to make quickly. So the course is a volunteer effort, so we are not paid for it at all. We have to fight very hard to get funding for just giving these courses. We believe in it, and so we did this, and we do it now so long that now slowly NSF and then NIH warm up to it, because they see how much good it does, and they give us the money. But anyway, so the money only goes into preparing the course material, but it doesn't pay our salary, and so we just do it because we really believe in it. So the hands-on sessions are really an important part. I mentioned this already, and you basically, the goal is that you learn to do computational biology. I like to put it sometimes a little bit differently. I like to say that there's a lot of knowledge in the world today, but very few skills. You know, the difference between knowledge and skill is knowledge, yeah, you learn it from the textbook, you can tell some numbers, but you cannot do anything with it. And the skill is that you have actually the ability to do something, to get something done. And that is what we try to emphasize a little bit, that at the end of this week, you might have a few more skills that you then can really do research that you couldn't do before. And so then I like to encourage you to help each other. That is good. And then I also very, very much like to encourage you to look at your own system. So I heard from almost everybody that they have their own research interests already, and if you are taking, you know, like 30% off here and get you the PDB of your own protein system, download it and apply it to the tools that we teach here, that would be totally good. And then if you are asking the instructors about your particular system, what you may need to do, or how what we tell you here relates to it, that would be just the right thing for you to do. Okay, then we also like to get feedback from you. So every day we will ask you to write out a sheet of paper to grade the individual components of the course, and then also make suggestions and give comments. That's very helpful for us to, over the time, improve the material. And that was very, very valuable. And so we ask you, giving us something back, to actually provide us with your comments. Okay, so the teaching assistant, they bear the brunt work here. And so here you have them, and they come from Pittsburgh and from Urbana, and there's a very, very nice group of people. So I think you will be in good hands. Okay, good. So then let us begin. And the first part is always a little difficult, because I talk about VMD, and many people think VMD is just a program to make pretty pictures. And so, you know, it's sort of a little embarrassing for me as a theoretical physicist. I'm a theoretical physicist to talk about making pretty pictures. But you will realize that VMD is much more than that, and so that I can stand in front of you with good conscience. So in a way, our view of both the use of VMD and the use of NMD is that of a computational microscope, that we have an opportunity by modeling systems, or by taking information data from the crystallographic databases and other databases, that we can visualize a system, model them, calculate their properties, calculate their dynamics, and thereby provide a view of these systems, a view often that is not provided at all by other microscopies, because you know, the scale that we are talking about is about you know, today typical a nanometer to 100 nanometer, is not covered well by light microscopy, since we have wet samples, also not so well by electron microscopy, definitely not by crystallography, because there you have a very special crystal in which you are taking the data, the crystal form with many contacts and so on. And NMR comes closest to the reality, but it cannot do big systems, and also it is pretty tough to apply in many cases, and so it is you know, hard method. So this computational microscope gives you views that can be extremely important. And you know, some people may say, "Oh, this is just computational. Ooh, I don't believe it, just virtual." I don't think that the computational microscope has more imaging artifacts than you find in all these other methods. You know, they all have their problems, and you have to be very careful. You have to be careful, and with the computational microscope, absolutely. But it's just more psychological that we are not. That we are maybe virtual, otherwise we are giving pretty good pictures nowadays. The picture that we give and that is shown on the left side, and that's sort of important, they go over a very broad scale of living systems, from the electronic all the way to the whole cell. So I will mainly show you things in between, and not so much the electronic, but I can tell you, we'll just show some very simple examples, that you can visualize electronic orbitals today in the protein, in DNA, and in situ, together with the structures, you see the electronic orbitals, and you can also visualize entire cells with their content that can be experimentally seen with electron microscopy, and then pieced together in a computational model. So that is basically, we cover all the bases that you need for living systems. Now, our microscope is made of two things. One, of knowledge. That is actually mainly the area of molecular dynamics, and so I will cover this more tomorrow, and I will be just very brief by telling you that we simulate macromolecule just like Boeing simulates the next generation of Dreamliner airplane. We simulate molecules. We have lots of knowledge. The knowledge comes mainly from chemistry, actually, about the chemical bonds and their physical properties. Physics, and knows about the forces and how the forces lead to motion. And mathematics gives us then the best strategies, computational strategies to have efficient algorithms. And then we have to put it into computers through software and translate that into programs and use strategies there to do it in parallel, in a parallel fashion. And then we have to then put these programs into real computers and use them effectively. So that is one part of the microscope. Maybe you can think of it the metal tube of the microscope. And the glass lenses is, so to speak, the VMD program. And the VMD program is very visual, of course, because it shows things. But in being visual, it's not like eye candy. Of course, it's no problem that it looks pretty, nothing wrong with that, but you have to appreciate that your visual brain is very, very highly evolved. So you don't learn it in school, although in some art schools you probably develop your visual brain, but normally you don't learn it. But what you have in your visual cortex here is a very impressive part of your brain. You can do great things about correlating information and classifying information and so on. And you do that all naturally. You don't learn it in school, but it is available there. And basically VMD appeals to that intelligence that is built into your brain. But then, of course, it also is on real physical characteristic, physical data. And here we have some examples. Here we see a buckyball vibrating. And here you see, we see that in VMD. That's, of course, pretty trivial to show. And here you see the electron orbitals as they move when the atoms are vibrating. So they are pi orbitals that are spread over the carbons of the buckyball. You see them here moving. This is the data here, quantum chemistry data come from Todd Martinez at Stanford. And so here you see a lipoprotein particle that scoops up lipids in the bloodstream and make them into particles and carries them then, for example, to the liver. And here you see about 300 ribosomes in a cell as seen in electron microscopy, in electron tomography it is called, because it looks like a patient in the hospital. It looks at tomographically at the content of a cell, and here we just see the content, which are the ribosomes, and here you see one ribosome, and we can resolve the ribosome, all these 300 ribosomes, every atom, and tell you what atom is where, and we can add many, many more particles than just a ribosome. So VMD can literally go from the electron all the way to very high content. And VMD can do today huge systems with several 100 million atoms, and so that looks a little mind-boggling, but we developed this over 30 years, and we are amazed ourselves that we can do such large systems today without problem. So we can really handle living system, not just at the level of a drug molecule and the level of a small protein, but really at the level of a whole system. So the most the characterization of VMD that I like most was given by the eminent biologist Carl Woese, and he loved to use VMD himself, and he called it a tool to think. And I think that's a very, very good statement. So you can do so many things with VMD, just like with your brain, and it also maybe is sort of like an encouragement to how you should go at using VMD and NMD. These are not tools that are push-button tools. You know, you put something in the computer, you push the button, and then it runs. That is, we call those students in the group. Hopefully, I don't have too many. And the nasty graduate students call those students in the group who push the button and then they go play ping pong or something. They call them monkeys. They push the button. You know, it's really a way to think about the system. It helps you to think about the system. You must have a hypothesis before you even start the simulation. You must have a clear question, and then you address that question with the simulation. And I will answer many of your questions very likely, because that happens all the time, by saying, you know, let's think about it. Or use your brain. I don't tell you, oh, here I have a trick like a recipe, like a cooking recipe, this recipe and that recipe. No, 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 I tell you, think about it. Use your brain. That is the best advice I can give you all the time, rather than that you imagine that there is some, you know, particular trick, and when you know the trick, then everything works. However, I should tell you that sometimes there is actually a trick, or there is some knowledge that you don't have. So it's very important that you ask questions here, or maybe afterwards that you might want to go to the mail, what do you call it, mail VMD, and maybe they have an email club. I'm such an old guy that I don't even know the right name. How do you call these kind of things? And so there you find what people discuss, questions they discuss, or you ask your question and people help you. How do you call it? Forum? Yeah, a mail forum, but probably a very good name, yes. And so really try to find out, or maybe send us an email and ask a question. Be specific, then we can answer it usually very quickly, and that may save you, you know, 2 months, 3 months time, because you're just at some roadblock where you just need a little piece of information. But in general, don't assume this is here a bag of tricks that is all about, but it is really about your brain that you want to use. But you know, once in a while you need to know how to do the next thing. And here we see that we have many kinds of data that are actually being used and represented in VMD. One is just what you might first think of alone, that are atomic data that describe the structure and also the dynamics of macromolecules. You have a membrane, you have a channel, and you have your DNA that goes through this channel. And so these are sort of atomic level data. But we also have volumetric data, like quantum chemical orbitals of the electrons that make the macromolecules. Here we have density data from an electron microscope that sees a protein in a functional form, where the crystal, the red one, was not in a functional form, and then you can see that you can move the non-functional crystallographic form into the functional form as seen by the density of the electron microscope. And then we have sequence data, and we can align sequences, we can make phylogenetic trees. And the great thing about VMDs, you can have them all together. So with your molecular orbitals, you can put the atoms. When you have sequence data, you also have at the same time the structures. If you have many sequences, you superimpose all the structures. If you go to one particular point in the sequence, you see then that particular amino acid highlighted in your structure. And also the volumetric data you see here, they talk to the atomic level structural data. So the nice thing about VMD is that it has these different types of data, but they are interconnected. They are not separate into classes, and so that is very, very helpful. So so here we have then the view that we have also cell scale description of VMD, and so here we see like a membrane, and we see channels. In this case, they are mechanosensitive channels that are preventing osmotic shock of bacteria. And you see they have a filter in front of the channel that sits in the membrane. And the ions go through, and they selected little windows and let it only go through. So you have views here of the entire pretty large scale view of the cell, but that is nothing compared to what you can really do nowadays with VMD. And so just as a little entertainment, I might now show you something more. So what I first do is I show you an absolutely lovely book that is available, unfortunately, only on Macs. It's an iBook book, and they only sell it for iBook because Apple. Why didn't we move it here? Apple is funded the book, and so the book costs only $1.99. It's called Life on Earth, and written by the main author is the very famous Harvard biologist E.O. Wilson. And so now I show you a video from this book.
Flexible filaments that form cross-linked bundles.
Sorry, I have to go to the beginning. So.
The cytoskeleton is a dynamic network of filamentous.
So there is some narration and also some sound. The music claims it knows the sound in the cell, but here you see a lot of striation in the cell, and networks, for example, of actins and microtubules that are giving mechanical strength to the cell, and they grow in the cell to push amoeba forwards, but to also provide a street for motor proteins. And so this is in this textbook. It's really a great book. It's just sort of like being written now. It is about maybe 1/3 finished. And you know, you see here these molecules, but it's a little bit Hollywood. It's they actually spent a lot of money on it, particularly at Harvard University, visualizing it. But this in detail, if you look at it, so I say two things. One thing, I say, oh, it's fantastic. Real, you see how things are in the cell. But then when I look in detail, I say, oh, there's something wrong, there's something wrong. More important is that we see the whole view of the cell. This is where biology is going. But you know, in detail, one saying, oh, I want to get it scientifically right. And that is what actually VMD can do. And so I would like to show you the very latest that we did ourselves in this department. And so here is the answer of VMD to this kind of picture. So basically we do the same. We think it's great. And this is about a photosynthetic organism, a whole organelle in the cell that is described at the atomic level with molecular dynamics and VMD. So here's a photosynthetic cell. It has many of these rings. This is a slice of the cell, only 200 angstrom thick. Through ion beam milling, the cell was sliced and then viewed with electron microscopy. And now you see all these spheres, and here's one of the spheres filled now with an object that is in these spheres, and it's called a chromatophore. It's called a photosynthetic chromatophore. It absorbs light and uses the light energy to synthesize ATP. And this is what each of these spheres does by itself. And here you see it's made of 100 proteins and 6 different kinds of protein. Here you see the inside of the protein, what the protein is. There are many chlorophylls and carotenoids in the proteins, and they absorb the light, and then they sense excitation through the system. Here you see an ATP synthase, and this is all correct. You know, here we don't show every atom, because if we would show every atom all the time, we wouldn't see anything. So we just simplified the moment through surfaces. But if I would go anywhere, any place else in the movie by just maybe trying to catch it here, and then I go maybe somewhere where here, this would be a moment where you see now one of these light harvesting, one of these photosynthetic proteins called light harvesting complex 2. You see it has 27 chlorophylls, it has 9 carotenoids, You see every correct atom. Again, we took put away many atoms so that you see the most important things. But you know, you have here the same kind of level, cell level picture, but at every atom. And that's what VMD can do. So I told you before, we can put it all together, and here you see for the first time that it's actually really working. So this whole chromatophore that you saw was actually the biggest simulation ever done today on these huge computers that are available today. It's a 100 million atom simulation where we let all the lipids and all the proteins of this thing move and equilibrate together and so on. Okay, so you see that literally VMD can go all the way from the electronic degrees of freedom, which play a very important role in case of a photosynthetic system, all the way to the, not here quite cell, but cell organelle. So it's really like a really important piece of the cell level system that can be also captured in VMD and also in MD and described at the level. And that is sort of little bit the future of the field that goes more towards a system level more and more, and doesn't just stay around the single molecule level. Okay, but that's now the grand vision, and now we want to go back to the daily grind. And so here we have now the key components of VMD. We have here our graphics window that shows the protein, and then we have here the main menu. This is actually what you see all the time. First, when you start VMD, the first thing you see, you're getting a graphics window, you're getting the main menu. And from the main menu, you can dial then other things. Most of the time you dial the graphical representation that permits you to control the graphics window. You are getting also actually immediately delivered a window that gives you all the outputs of the program, but you can also put input there, so that is just your normal C window, C language window, where you're getting your Unix window where you put in your Unix commands. And then you have also a special command window where you can put your scripting, and so this is the TK console. So you have the main menu where you can choose everything. Most of the time you choose a graphics representation to sort of alter your graphics and to analyze certain physical characteristics, but there are many other things that you can choose here. We will see that a little bit. Then you're getting a Unix window there where the program talks to you, but you can also put certain commands back. And then you have a window where you can do your scripting. And you will become familiar through the tutorial with these windows. And so now I want to immediately begin with an example. And the example is chosen not about, here's a molecule that we want to look at with VMD, but it's also chosen that I need to tell you how we look at things with VMD. Because we don't look at VMD to just enjoy our eyes, to make pretty pictures. We are scientists. We use it as scientists. And that I want to immediately tell you with this example. So this is a very, very famous protein. The name indicates already. It's called ubiquitin. It has 76 amino acids, and it's called ubiquitin because it is ubiquitous. And you find it in most primitive cells and the most advanced cells. And what it does, it labels proteins according to where they should be localized in the cell, where they should be going in the cell. Some proteins should go into the nucleus, some proteins should be excreted, other proteins should get the kiss of death label. They should be destroyed because they're misfolded, and so on and so on. So you need these address proteins, and here you see a little bit of a closer view of them. when you have a protein that you want to localize in the cell, then if you would only put one ubiquitin, you have only one address label. That means they would go all to the same place. That's not good. So you actually can now write with these ubiquitins several different addresses by linking the ubiquitins together. You link the first ubiquitin, and then very often you link actually 4 ubiquitins, but you can also have smaller or larger numbers. And they are now linked together. And the way they're linked together is that the ubiquitin has a C-terminal tail that sticks pretty far out. And the last amino acid can be connected not only to the protein, but also to one of these ubiquitins. So one ubiquitin holds with the C-terminus the next ubiquitin, and that one holds the next one and the next one. And now they can hold each other on the left shoulder, on the right shoulder, on the hip, and those places are identified through lysine residues. And they can either be connected through lysine 48, or through lysine 63, or through lysine 29. And depending on if you connect like this or like this, you are forming a very fat little label, or a very thin label, or a very rigid label. And thereby the physical property of the label, you know the address. And so that is basically the idea of this protein, that it can make little pieces of strings that are, through their physical characteristics, acting as address labels. And so now when we are looking at ubiquitin, this is a picture we have in mind. We have a certain picture now we want to say, oh, how will that work? Not that we just say, oh, no, let's look at ubiquitin. Don't tell me anything about it. I just want to make a pretty picture. No, you should have really some preconceived notion. Don't be too stubborn with it, maybe be open-minded. Maybe your notion is wrong. But you need to have some kind of question, some kind of idea, something you want to explore, something specific you want to explore. Okay, so that is what we do. Now we use the program. Okay, so let's now. I pre-stored it, but I want to sort of reset the program to state that. Okay, so now I will take everything away. And so I put it in a form the way you see it when you're loading the program. How you load the program is very trivial, and we don't need to do that. So this is how, when you are loading the program, this is what you see, and so this is what you see. So you load it, somebody said to you, oh, I have a file with ubiquitin. This is how it looks like. Okay, good. What do we have here? Now here you see already that VMD starts when you put it here. Maybe I get a little closer by scaling it up. It has now lines, and this is a representation that you are getting, and mainly for the reason it's the cheapest representation. So if somebody gives you a gigantic protein or whatever, that your computer doesn't freeze because it's too complicated to draw all the detail. With lines, it's just a picosecond. But you don't see much. So what we can do is instead of lines, we go to Van der Waals spheres. So now we have this one. And so now you see that I scale it a little bit back so that it doesn't get cut away. So now you see what we have here. And so now I happen to know that I can look at these little red balls there, and I look at one of them, and this is good. I just look at one of them, and there we have one. If I put an equal sign, then the ball is in the middle. Now it's a little bit too big, so I have to make it small. And here we have this ball, and now if I rotate it around, then you see it's a water molecule. you see that the water molecules look a little primitive. It's not nicely done, but we have here so many water molecules that VMD is not doing a very accurate a picture of it. Here we have the resolution of the spheres, and if I now go, I can make it real neat sphere. But if I would do that for all the water molecules, it would be too expensive. And so, you know, it's then for the many water molecules, it's stupid, but if you have one, you can do it very, very nice. So I go better back to what I had before, which was just 12, and that it can handle even have that many water molecules. So you see we have obviously there water molecules, and so now we are looking at all of them again. And so here they are. Oops, I did something wrong. Yeah, okay, so when you do a typo, it immediately tells you, doesn't understand you. I do the equal sign, then I'm getting my standard view. But you know, I'm not interested in seeing the water molecules. I want to look at ubiquitin. And now you have this very powerful atom selection language in this selected atoms line, which you will realize it understands Boolean logic. It's very, very powerful. In this particular case, it's very simple. I just say, I don't want to see everything. I just want to see protein. So I write protein, and now I just see the protein. Now the protein is not very illuminating this way. I scale it up a little bit. But you know, we see all the atoms, and you know, you don't learn much about the protein by looking at the atoms. And so now VMD has many ways of representing this protein, and then to sort of understand its function better. And the first is you would like to not see every atom, you want to see the architecture of the protein. And so the best way of looking at that is something that is called new ribbons. That's my favorite. No, not new ribbons, no. New, sorry, oops. Not new ribbons, new cartoon. That's absolutely my absolute favorite presentation in any case, because it gives you a good overview of this protein. It is colored now one color, and we use now a coloring method by secondary structure, because then we see immediately what this picture tells us. And I now scale it up a little bit so that we really all see it very well. And so I can also translate it by around if I want, and then I can rotate it again. And now you see that we have different colored elements. The purple is actually an alpha helix. So you see that is clearly some helical form there. The yellow one is a beta sheet. They are beta strands, parallel beta strands, or rather anti-parallel beta strands. And then we have the blue ones. Those are loops, but ordered loops. They have a particular structure. And then we have white areas. They are like, for example, here at the end, they are disordered loops. They also look like the blue ones, but the blue are, even though they look sort of rather arbitrary, they're actually structurally sound, whereas the white ones are not structurally sound. They are sort of very floppy. So now you see very, very quickly this molecule. Now, rather than using any of these ones, I just delete them all, so that I follow like what you would do. Now I deleted one too many. Let's see if I got it again. Good, great. Okay, so now we want to learn more about the molecule, and you can actually do more than one representation. You can duplicate this representation, and now you can instead of looking at this one, you can have a second representation on top of the other ones, where you, for example, see only particular amino acids. I put here res ID 1, 2, 3, 4, 5. These would be the first residues with ID 1, 2, 3, 4, 5. And I put them here. And now I don't see anything because it's the same kind of representation on top of each other. But now I give it a new drawing method. I draw it like licorice. Licorice is my favorite representation if I just want to see protein residues. Now there are also these water molecules. They also have residue numbers. So I write protein and res ID. And now I see only the protein parts and not the others. And so now I see there in these colors, I see something else. Now I can do my first representation, that new cartoon. I can click it away by double clicking, and now I have only the new ones. And the new ones, I don't want a color by licorice, but rather by residue name. I thought, oops, what did I? Oh, I did it wrong here. I want to do this one by secondary structure. I was in the wrong department. So here I want to color my name. And so here we have here, and somehow I had did something odd. Okay, I have a very, very, no, something very odd here. Okay, I live with it now because it's so. Okay, so here I have now my molecule. The first 5 amino acids, because I wrote a res ID 1, 2, 3, 4, 5. And now if I don't know, many of you, particularly the chemists, recognize already the amino acids, but for the rest of us, we could add labels to the amino acids by going here with the mouse to label, and then we say atoms. And now if I now hit on these amino acids, then I will be getting a label next to them. And if I go to my about a mistake when I okay, so the label is not easy to see, but in the window it would have written it out to me, but I cannot see it. So I go now here, and so this label it tells me that this is a methionine. So it is the first amino acid very often, and it is so that it tells me it's a hydrogen atom. If I would click on the yellow one, it would tell me it is a sulfur. So if I click on this one, what is oh, it's off again. So there it would tell me it's a sulfur. So then if I would go, oh, sorry, I also do a little differently. I go like this, rotate it like this. So here I have it in front of my eye here. Okay, so I have the first one, and now I go to the second one. I click here. Again, I lost the label. And so if I click here, then it tells me it's a glycine. It's an oxygen of a glycine. So the second amino acid is a glycine. If I go here, then I would read, if I read the label, I would see it is an isoleucine. And if I go here, then I see it's a phenyl ring. So if I go here, phenyl, and if I go here, then it's a valine group. If you would look in detail, that it's a little bit hard for you to see. But you see, you can go through the protein, amino acid by amino acid, and see what it is made of. And now we can add this representation back to the other one. You see they're on top of each other. We know where everything is. Okay, so that is, we can have two representations. Now this new representation looks a little silly, so I take it away. And I also make the protein a little bit smaller again. And I also get rid of the labels. That's a way of doing that by going here to labels and tell it to delete all the labels. So I now delete it. I scale it up a little bit, and so this is now my new protein. And now, so this is one way of looking at it, but we see only the architecture, the sort of like the key idea of it. Now we would like also see the physical characteristic of it. And that you do by looking at the surface of the protein. So what we are doing for that purpose is we are going here to this, the most important graphical representation menu, and we have now a second, we make now a new representation. So here you go, a new representation. And this representation, there we want to show the surface. So we go now here and go here, and now we see the surface of the protein. Now the surface, we see the secondary structure, because the color that we chose was by secondary structure. That's not so illuminating on the surface. What's illuminating on the surface are the amino acid types. So if we first put the amino acid names there, the 20 amino acids, then it looks like pretty nicely colored. But again, it's not really too meaningful. We see that the 20 amino acids, we just look in a color table, which one is which. The best way of coloring on the surface actually by residue type. And now the blue ones are the residues that are positive, the red are negative, the green are polar, they like water, and the white are non-polar, they don't like water. And so now we see immediately where we have positive amino acids, where we have negative amino acids, where we have polar amino acids, where we have non-polar. And so it's very interesting that you look at it and you see, oh my God, there's so much non-polar, but this is in water. How can it be so non-polar? So you see that the green is polar, and the charged amino acid, of course, they also love water. Why is there so much white? Any answer? So now we begin to think about this thing. And I told you before, you have to think about what I told you before, and now we have this one. It's maybe not so trivial, but you can, in principle, you can find it out. Why do you think that someone non-polar? So it's non-polar because there are more than when one ubiquitin. I told you usually 4. And so when the 4 come, what they do is they get together. They have to have a certain way to get together. They need a certain adhesiveness somewhere. And that's what proteins in water often do is they adhere to each other with the non-polar elements, because water doesn't like it, but the two non-polar like each other, and so it's like a glue. And so these lots of white tells you immediately, oh, this protein is probably not a loner. It has partners, and in this case, the partners are the other ubiquitins. And so we often have 4, and they will touch each other such that they are trying to touch each other with the non-polar surface, so that at the end, the stuff that is exposed to the solvent in the cell is all polar. Very, very nice thing already. Okay, but now we would like to see these connections. So I told you already that the last amino acid is the one that is connecting them, and this protein has 76 amino acids. So I put here now 76. Now I don't see 76 here because I chose a representation. I take everything away. I see. This is 76. It's too small. It would disappear there. So what I do is I make it van der Waals. And now I blow it up. That become real fat. Oh no, not sphere resolution. So I pushed the wrong thing. I blow up the sphere, so it becomes an amino acid on steroids. And now I color it here's a coloring method, and we use the colors by recognizing what atoms are involved or what type of amino acid we have. And now we just choose it now to have a signal. We want to signal something. This is the last amino acid of this protein. So we go to color ID, and here we have then many different choices of color, and then let's choose one color that is maybe not appearing there very often. This is actually what I'd shown before here when I was sitting in my hotel room. This cyan is not used by VMD to signify any other properties. So now we have it, and now we add to our protein the surface, and now you see here where we have this last amino acid. That is where one of them sticks to the next one, and they stick now to the lysines. I told you on this slide I had, you said they connect to lysine 48, through lysine 29, or through lysine 63. I just memorize those numbers. So we want to see those lysines. Now we do that one by using one of the many tools, over 100 tools that we have in VMD that are called plugins or extensions. And here we go to this, it's called analysis, where we have many tools. And one of the tools is that we can have a sequence viewer where we see the sequence. Where is it now? Ah, here, there it is. So, that was a problem. I showed everything before and I screwed it up a little bit that way. So here's a sequence viewer where we see all the amino acids of this protein listed here. So now we find our 3 lysines. So we go here to lysine 29, and we want to find it. We don't see anything. Why not? Because it's covered by what we have here. Now what we do is we do everything away, except when we hit this thing here, we are getting a new representation to which amino acid we select here in the sequence viewer. And so now we want to look at lysine 29. There's lysine 29. For some reason, the sequence viewer has a real ugly representation of the lysine. So we are doing again van der Waals, that we see them well. So we choose now, we are going here. And so we don't do bonds, but we rather do van der Waals. We are also choosing the same color ID that we had before, namely this cyan, that we can recognize it nicely. We also do the sphere bigger, so that we recognize it, that it shows big. Okay, great. And now actually maybe we don't have to make it quite that big. And now we add the surface back. And we add also this first one. And now you see here's the first the last amino acid, the C terminus. And here you see one lysine 29. And now we are going to the sequence viewer, and we can also show the other lysines. Here we add lysine 48 by shift clicking. Then we see lysine 48, and by shift clicking on 63, we see lysine 63. And so now you see the 3 lysines, and you see that there are different places on the surface. And now if we are taking one of these guys and connect it with their C terminus to one of these 3, you can easily imagine that you are getting now a series, like a necklace, that is different depending on if we are adding this one to this position, or to that position, or to this position. And so here, this was a very trivial case. But what I want to tell you is, never use VMD half drunk or tired or something. Ask yourself a question, explore with it. Have a little dialogue with yourself, and that is how this program is useful. It's not useful just if you go around and look a little pretty here and maybe want to impress your boyfriend or your girlfriend or whatever. Forget it. It's really a tool to see. You should have a purpose. Okay, good. So that was maybe very long, but important. The key message is always ask a question when you are going to VMD. If you don't have a VMD, have a cup of coffee, think a little bit about, take a little nap, come back with a question, and then start it. Okay, so now let me continue then. So, how are we? Do we have a coffee plan? Do you remember when it is about? So we will maybe do a little bit more, and then we have our coffee time. Okay, so now we have something that that we call tutorials. I introduced that already. That's what we are working through in the afternoon. And tutorials is basically, they have also some examples of molecules, but there the program is the most important thing, not the molecules that you are dealing with, that you're encountering there. So that's a tutorial. But then we have also something that we call case studies, where it's the opposite. Now we think that we know the molecule, and now let's see what we can, that we know VMD, and now we say, what kind of explorations can we do with VMD? Just what I told you now, ubiquitin. And so there we have, I think now 12 or maybe already more case studies, where you just really now explore different molecular systems, start with something simple as water and ice, and goes all the photosynthetic protein, things that interest you, so it goes all the way there, and many things in between. And so there you see the act of exploration. You know, you look at certain physical characteristics, and so you learn to explore with VMD, and so there the biology is more important than actually which button you should push. And so we have also one case study on ubiquitin, but if you go to our website, you find that we have quite a number of these case studies, and we are adding to them every year. We're adding 2 or 3 more case studies. Okay, so I mentioned already that we have also sequence information in VMD, so that will not be covered. Except I think with the last day there will be a little bit of sequence analysis in this course, but so you can do a lot with VMD. Maybe they actually use on the last day also VMD, and in VMD we have the sequence viewer that I used already. That's just a very primitive tool that just allows you to look at the sequence of the protein that you have here. But then we have a very powerful tool that is called multisec. Multisec comes from multiple sequence. It can do multiple sequence alignments. And so here we have, for example, a situation that we have sequences of related proteins. And so here, I don't know, maybe 7. And here we see also the structures of that correspond to these sequences superimposed. You see some part where the sequence is superimposed very, very nicely. In other parts, there's more variation in structure space. And in sequence, you see then conserved amino acids, and you see deletion, and you see whatever you can do in sequence analysis. But multisec can do more. Multisec is also very powerful in making what are called phylogenetic trees, the family trees of proteins. So if you have certain proteins, like they're all written here in the protein in the sequence database, then they are related to each other. Some of them are very similar, very homologous, so they are then brothers and sisters, so to speak. And then others are more related. They are sort of cousins or distant cousins. And then some of them are even further related with each other. And you call this kind of family tree a phylogenetic tree. And that tells you basically how the proteins evolved. And there's a lot of wisdom in the evolution of the protein. So if you look at protein function, you learn a lot from the evolution that is hidden behind these phylogenetic trees. Here you see the protein superimposed. And now if you are going in multisec, you're going, for example, to this point, then you are finding immediately in this superimposed structure what change was made that this protein went here and this family of protein went here. There was one or maybe two amino acid substitutions that happened at that moment, and now you're getting two new families. And then here you see you're getting another separation, another separation. All these changes, you can, by just clicking on them, you can see, and you can also shift click so that you see these changes and these changes and these changes, and let's say, you know, these changes here all together, that you see along which path one particular class of proteins evolved. So this is really a very powerful tool to get these kind of analysis into phylogenetic trees, and then you have this great way of looking in structure space what actually happened in evolution. You just don't see, oh yes, there was a mutation, but you have no clue where it is in the structure. And once you see the structure, of course, you also have often an idea what was the functional relevance of this mutation. You see that then right away in VMD. So this is a very, very important, powerful tool that you should familiarize yourself with. So like physicists, they all think they are very smart, and they look at a protein and say, oh, I look at it and stare at it, and then I should find out, you know, how it works. And the biologists are really smarter. The biologists, they say, yeah, I'm pretty stupid. So let me look at the sequence of these proteins. Look at my protein, but also at related protein. And then look which amino acids are conserved. And they say, ah, here 20 amino acids that are always conserved. And they must be important. And right away they see what's important. Whereas the smart physicists, you know, they, I just use my brain. They have, you know, to deal with 300 amino acids, and they're all over the map, and much harder to find out what is really key. And so to use this kind of conserved amino acids, to use the evolution that led to a particular protein is extremely helpful. And so that is what biologists learned, that the respect for life on Earth, how it evolved, and what has been learned from that, and utilize that information in understanding life today. Okay, so then I said also that VMD can handle volumetric data. This is already something that I showed earlier, the buckyball, and then superimpose on it the electronic orbitals. And here you see actually something that's very, very common activity with VMD and NMD, and that is the interpretation of electron microscopy densities. Here you see a protein, a part of a ribosome, that is put into the density that is measured by electron microscopy. You see then, initially it was ticked out, and now when the electron microscopy measures it, it finds that this is where the density is. What happened is that this protein was wrong, quote, because of a crystal form. Here you see how the transfer RNAs are reading here the codon. Here's a codon, and here's the anti-codon loop of the tRNA. Here's the messenger RNA, is not shown here. And you see here how the loop goes around. Here you see also a nice comparison of how well once you interpreted your structure in terms of the density, how it all fits together well. So this kind of interpretation of electron microscopy data through simulation, where simulation helps you to move the density into the electron microscopy observed density, that's a very, very common thing you do with NMD and VMD. And there it is very important that VMD can actually show you both density and structure. So also VMD, also that is an understatement of the year, I would say that's the most important function of VMD today. It can determine the physical properties of molecules. So like, for example, it can show you the hydrogen bonds. It can show you salt bridges where amino acids of different charges interact with each other. It can measure distances. It can determine surfaces, exposed surfaces, so-called SASAs. It can show you the electrostatic field around a protein by calculating the electrostatic field. So you can study many physical properties. So if you have a structure from the protein database, or you're doing molecular dynamic simulation that shows that actually there is an event that changes the conformation of a protein, then VMD can now tell you what kind of physical properties change along with this conformational state. And so here you just see a little introduction to what kind of calculation you do. Here you see a list of them, but we have a much bigger list. And, oh wait, that's not here. Sorry. We have a much bigger list that I thought it would be coming now. So there are basically, I think, almost 200, they're called plugins now, where you can calculate certain properties by invoking scripts for VMD that have been developed by other researchers. So the way we handle VMD is that when it actually started, the development of the program started with a revolution in my group. So we had we were doing molecular dynamics, and we had, just like in any other group, we had one graduate student wrote a program, and that was then inherited to the next graduate student. And the next graduate student went all bananas because they didn't, there was not enough documentation, there was no commentary, and the graduate student was long gone. And so this happened, you know, from generation to generation. And one day the students came and said, Klaus, we had it. We don't do it like this anymore. We write now a completely new code. And that code is first not using Fortran or some arcane language. We write it in C and C++. And also we have a condition, we make it freely available, free software. So there came the revolution, and they thought I would say no. And they probably thought they are safe, that they can make a suggestion and I say no, and then they could say, oh, we told you so, Klaus. You didn't listen to us 10 years ago. But I was very happy that they made a revolution. I said, great, fantastic. I help you as much as I can. And so then they really got together, and it was the students who started these programs. Yeah, the professor, the only thing I did was that I wasn't in their way, and I also helped them as much as I could, but you know, more like cleaning the floor or getting enough Coke or something, but not you know, telling them do this, do that. And so one of their conditions was, that was like some time ago where this was sort of much discussed, is that the software should be free. And so we made it free software. And the great advantage of it, that was clear to us then, and it turned out to be completely true, is that since it's free, many people will be very happy that they can use it, and they will also be happy to contribute to it. And so today we have really many, many contributions to VMD and MD. Some of it is in forms of scripts that are the easiest. You can, if they send us a script within you know, a day, we can integrate it in the distribution of the program. But we also sometimes, they are so important, or there are scripts that are too slow to do real jobs, scripts running more slowly than the core code of a program. And then we're putting those scripts into the program as core code, and then they run much, much faster. And so we have both models, but VMD and MD are programs today that got many contributions from other users. And so that is really great. And so particularly these kind of properties are being used that way. And today, when we run molecular dynamics simulations at the big computer centers, then 50% of the actual computer resources go into the analysis with VMD. So we are running, for example, on the Blue Waters computer, one of the biggest computers in the world, very big simulations. And then after we are done, we need 50% more time to actually analyze the results. And so that is really very typical. I would say that ratio will only increase, become maybe 100% or more in the future. And so you should be aware that VMD is really a program for characterizing the physical properties of molecules. And so now I want to show you this particular. I want to also show you some other aspect of VMD, and that is that VMD is completely natural in analyzing dynamic data. So if you make a pretty picture with VMD of one conformation of a molecule, like for example a folding molecule, you can now use all this prettiness, this choice of colors and that choice of property along the entire trajectory. And so this is what you see here, and that is what I want to show you next. So now we have just maybe 10 more minutes, but that are fun more minutes. So here you see VMD running, and then you see here also, I will show it to you in a second, we call a timeline where you see the properties of this run a long time. So here you see some numbers, and they are basically time slots. And now here you see the amino acids along this axis, and here the color tell you the secondary structure. The violet means alpha helical. And now you see that very quickly you're getting a lot of alpha helical content in this protein. You see here the helix, here the helix, here the helix, here the helix. So this is really what happens. The protein folded quickly to a certain degree of alpha helix. Okay, now let us look at this kind of situation. So I go out here, and now I start in this VMD program, I hope. Okay, great. So now I have to hide everything else. And so now I have just the same VMD that I had before. And now I will again sort of delete everything so that I have the starting point. So you have the starting point. And I go here, so, and now I have stored in VMD not just one structure, but a whole simulation of many structures, and this is of a folding protein. And so now I show you this. So here we have this one, and you see here the protein is initially stretched out. At the end it is folded. And here we see how it folds. Now you might get seasick from seeing these contortions. So this is actually not the best way of doing it. So what you do immediately is, in order to see a little bit more system there, so you go here to drawing that is for trajectories where you can low pass filter over several frames over a certain time. And if we are now looking at this trajectory, then you see we average always over 10 picosecond in this case, and then the motion becomes much more much smoother. We sort of average out some of the thermal jitter that you see there. Now you see really how the protein wrestles with itself and then folds finally into this last form. So if we keep it here, then we have it there. So now we could also say, so we had one particular coloration of the molecule, and this coloration is being kept through the entire trajectory. Now what we can do is look at the physical property of the molecule. So we create a new representation. Sorry, that was not what I wanted. Create a new representation, and in this representation, we look at the hydrogen atoms, at the hydrogen bonds, because we know that the helices are being stabilized through hydrogen bonds. So we want to see how hydrogen bonds develop over the course of the folding of the protein. We would like to color the hydrogen bonds. We use color ID to see it better. And we choose the color, blue is fine. We keep blue. Maybe just for the heck of it, since we are here trying out, we take an orange color. And now we can also choose what we call a hydrogen bond. There are some very precise geometrical conditions for this is a good hydrogen bond. The Prussian condition, very precise, but let's be a little bit looser because it's a folding protein, you know, like it slowly goes to hydrogen bond. So we are a little bit more generous by making the conditions a little wider than usual. Also the directional angle of it we make a little bit bigger, and then we draw really a fat line there so that we can recognize it. Okay, now we do the same. We just do our run, but now we hope that we are seeing actually the formation of the hydrogen bonds. And we do, except I chose a lousy color. I can see it, but you cannot see it. So I now have to stop it. And now I chose the worst possible color. I go back to blue. I see the blue already. You can see the image. So now we do it again. And now you will see better how they develop. I made another mistake. I have to average all properties over the trajectory, and so I have to here also choose 10 time windows for the hydrogen bonds, and they are much smoother, just like the rest of the first representation. So now it will be much more even. So now you see how the hydrogen bonds go, and you see really how they go nicely. And so now we can also do other things to this thing. So you see then if I now take it and go to the end, you see then we have it at the end. We can rotate it around. You see where the hydrogen bonds form. The hydrogen bonds that are in the middle there, they are between water molecules or other amino acids that are not shown directly. So now we can also add to this a surface representation. So I go here and I create a new representation. Now I say, oh, I would like to see the surface. So I go now and say, oh, draw me the surface. And I color now the surface by residue type, positive, negative, non-polar and polar. And I don't want to have the water molecules, I only want to have the protein shown here. And so good, that's what I do. And then and now I also have to be sure that I average the surface just like the other representations over 10 time windows. So I do this here. And now I can go back. And now we see the surface moving. So we go. But now it's a little bit silly. We see on the surface, but we don't see what's inside. And so this is something also really great about VMD. You can also look inside by making the surface transparent. So what I do now is I take, I go here to the surface, and now I do the draw style, and I have here the material of the surface opaque, I make it transparent. And since I'm a rich professor, I have a very expensive computer that had a very nice, expensive rendering method. Oh, I turned it on already, GLFL, good, that's good. And so now we are getting really beautiful surface, transparent, and now you see the grand finale, the protein folding, you see the surface, you see the hydrogen bond forming, you see the secondary structure inside forming, and so you see basically the whole picture. And so, and that is sort of like, you know, you once select your presentation, but it goes over the whole trajectory. And that is nice. You can even calculate along the trajectory, let's say the exposed surface and other physical characteristics. And that's what you can do. Now, some of these proteins, this is a protein that folds within about 10 microseconds. That's pretty fast folder, but some proteins fold only within a millisecond. So 10 microseconds, you know, for a while, but the millisecond is 100 times longer. So you wait here, this folds like a minute, the other one is 1.5 hours. That's totally boring kind of thing, you know, tedious, maybe impossible. If one folds in the computer, it can easily happen that the folding event or whatever is happening takes 2 days. And so what are you doing then? And so there the computer can help you. You can go to sleep and have a nice time. In the morning you wake up and the computer tells you, provides you all the data that you need in order to understand what happens. And that is what I want to show you, and then when we have our break. So now I go back to my presentation. And go here. And now, so now we have here our protein that we want to look at. And here we have now the information how this structure looked at as a function of time. And here you see the information for the amino acids, and here you see time. And in this particular case, you see that the secondary structure disappears. Why that? In this case, we break the protein, we pull it apart, rather than let it fall together. So you can do either thing. This is actually very, very often done in an atomic force microscope. So it's a very important computer experiment and real physical biophysics experiment. Okay, so here we have time. Here we have our amino acids. And here you see that we have plotted this. And this is the initial structure of the protein. This dotted line said, oh, we are here on this data set at the very beginning. And in red, we indicate one amino acids of these. And we show it. So it's already nice. We see the initial one and we see the one amino acid. And now we want to go along and see how this data set can be viewed next morning when you get up well rested. And so now we put the protein in the corner. And now we want to, so this is the beginning, just what we had before. And now we want to slide along the data set. And so now we go with our mouse and we pull it along, and you see now we get the time slices as we go along. And you see, oh, here's a protein folded in the computer. Yoo-hoo, I was sleeping and everything went just the way I expected. And now you can slice back to see, you know, how is it looking, you know, when you go reverse in time, yes, and so you see it. And so you see, oh, good, the data set is good, and I can now explore a little bit more. So now we do it a little bigger, and now we move a little bit around there. So now you see that we can move this one. This is a real VND window. We can rotate it, and now we can expand that we see every amino acid in this region around there. You know, this is a pretty big protein. There's so many amino acids that we cannot list all of them. And now we go along there, and now you go somewhere where we just lost some secondary structure element, and now we see there we put now the thing there, and you see now we are moving here. We look at this amino acid, and we go back as this piece of structure is formed. This is sort of like a loop that is formed here, and we see exactly we can glide along, and we can look at every piece in this sort of data set where we see what is happening. We can look at it and glide through the system. So it's a very powerful tool when we are talking about, you know, the analysis of your trajectory. Here we just look at secondary structure. You can look at electrostatic properties, at hydrogen bonding frequencies, at many properties as a function of time, and look at them. So if we now go to the next installation here, then we see just a little bit more of the same. You know, we can. You see as you go through, you can look at it, and you go here. And I think now we just push one time the amino acid through the protein. We just slide one time through, and we can go all along there. And so basically every point in this diagram we can use. In this case, it's already a pretty big protein, so we would be glad, but this can be done when it's a very long trajectory. Every time we see something interesting happening here, we can zoom there. You saw here we have not a very coarse view. We can zoom in real detail, see every little event, and then we can click there and see in the structure, we know what was happening. So this way we can look at the data very easily and very systematically without having to pay attention everywhere and stay awake all the time and so on. Okay, that's maybe a good moment to end, where basically, 1 second, where we basically, I want to stress that VMD, so that's a message for you, is you know, I said before, it's a tool to think and calculate physical properties. It's also a program that does everything, every graphics and physical analysis, not only for one conformation, but for whole trajectory. So you can apply to the output of molecular, of the NAMD program, and look how things change in time. You just have to choose one time your presentation, you apply to the trajectory, it goes all the way through, no problem, and you can even store the data in a way that you can easily find events to post-analyze.
Klaus, definitely this is a great tool, but I just wonder for this Titan simulation trajectory, it's starting from a very nicely folded, defined structure, and by end of the simulation, it seems like completely unfold.
Yes,
I
yes.
just wonder, you know, how to explain this. I mean, it's unfolded.
Yeah, no. That what happened is, this is an experiment, an atomic force microscope experiment, where you take the Titan. This is a Titan domain I-29, and you pulled it, and you can see in the experiment how you pull it, and you see how much force you need as you stretch it out, and you can simulate the same thing. So this is actually a muscle protein. In muscle, you have a lot of mechanical action, and the muscle protein provides a lot of elasticity to the muscle, that your muscle can extend and contract, but at the same time, they give it stability, structural stability. But you can sometimes even rupture a structure. It doesn't break apart, but it unfolds. And for example, you say, I overstretched my muscle, very painful, but it takes like a week, 2-3 weeks, and then it's okay again. Whereas if you rupture your muscle, can also happen, very, very bad accident, then it takes a long time to heal. And so these proteins, they are structural proteins, are very elastic, and like in flight muscle of insects, they go like real fast. And when you see an electron micrograph of it, you won't believe that muscle looks almost like it's breaking apart any moment. And there are then these proteins that are always keeping up, refolding, folding, and they're doing exactly this kind of analysis, and the proteins are being made to really stretch out far when they break, to give you a lot of slack, and then they revel together again.
So in other words, for this simulation, you add a force to
yes,
make it
there was the force added,
pattern.
exactly.
Yes, but for the previous, I mean, I'm just very
The
curious.
previous one, exactly, you're right. I should have.
Right?
There was no force. You just stretch the protein, and then you let it revel by itself, by just the forces, by the natural forces that are in the molecular mixed force field.
Yeah, it's folded in a beautiful shape, but I just wonder, you know, for that simulated folded structure, any experimental data to confirm that, like NMR or whatever?
Yes. Maybe it didn't. I think that even you saw it on that movie. There is at least maybe I should bring it tomorrow. So it folds to the crystal structure. So you basically, we have in the background, we have a shaded structure, and now we fold it out, then it goes, and then it goes right to the crystal structure.
How long for the simulation?
So this simulation, this was the Willen headpiece, was the fastest fall, about 10 microseconds. So that's a very fast one, and today one can fold proteins up to millisecond. That is actually this Shaw guy. He has a special computer. He can do milliseconds. We can do simulations about 100 microseconds, and then we peter out. Actually, the Shaw computer is also available here in Pittsburgh. Shaw donated one of his computers to the University of Pittsburgh Computer Center, and users can use it there. And so there you can get 40 microseconds a year. You can do simulations.
Very impressive.
Okay. So let's have a little break.